Hello, my name is Thierry Tonon. I'm a lecturer at the University of York. I'm lecturing in algal biology. And in the context of the SARTRAC project, I'm co-leading a work package looking at the composition and the reuse of sargassum. So I'm going to start by explaining what sargassum is, and then I will give you some ideas about how sargassum and how algae are used in general, and then also some ideas about how algae need to be processed to have some application in different kind of industries or in different topics. So first, I wanted to introduce what algae are, and in sargassum in particular. So you probably know that there are different kinds of living organisms. There are viruses, bacteria, fungi, plants, animals, and algae. Algae are a specific kind of organisms. And among algae, you have the microalgae, so the very tiny one, and you have the bigger one that we will call macroalgae or seaweeds. And sargassum is a seaweed. And there are three types of seaweed, and they are differentiated according to their color. Some are green, some are red, and others are brown. And sargassum is a brown seaweed. And you can find brown seaweed on the shore in many parts of the world, including in the UK. And what is interesting with sargassum is that most of the seaweed, to grow, they need to be attached. But sargassum, the sargassum we are working on, called pelagic sargassum, is a sargassum that is going to grow floating. It doesn't have to be attached to be able to grow. And in terms of application, seaweed have been used for centuries by us for providing mainly foods or providing any kind of other products used in different kind of industry. And some example is that we are eating algae and seaweed in particular. Our pets are also eating algae because there are some algae and seaweed in particular in toothpaste, in chewing gum, as wound dressing. We can also use seaweed to produce compost or to produce fertilizer. So we are going to use that for gardening, for instance. And seaweed have also been considered for the production of bioenergy, like liquid fuel for transportation or also for the production of gas. And there are also new applications which are developed from brown seaweed in particular, and this is the production of biomaterials. For example, if you want to do 3D bioprinting, you can use some compounds extracted from alginates. You can also produce paper and plastic from brown seaweed. This is what we call biopackaging, and this is a way of replacing the plastic which is derived from petrol. And another more, I think, recent application is the use of seaweed to produce fibers. The fiber that you are going to use to make clothes. And for example, it's a way of replacing cottons to make clothes. And why are we using seaweed and brown seaweed in particular? It's because they are containing some very interesting compounds. There are compounds that we can find in other type of organisms, such as a plant, for example. They will contain sugars, lipids, proteins, vitamins. But for instance, some of these sugars, they are specific to brown seaweed. It means that you can find them only in brown seaweed. And we need to know about the composition of the seaweed to know exactly what we can do with this seaweed. And this is what I have been working on in the context of the SARTRAC project. So we have been looking at the quantity of sugar, lipids, proteins, vitamin into sargassum. And for that, we are using different kind of techniques. I will say biology-based or chemistry-based techniques to look at the composition. And it can be techniques relying on change of color when you are mixing seaweed compounds with different kind of reagents. It can be more sophisticated techniques such as using gas chromatography or liquid chromatography, or also the use of enzyme, or what we will call also biocatalyst, to try to do some extraction from seaweed. And So I said that there are some interesting compounds and good compounds from seaweed, but there are also bad compounds. And when I say bad compounds, I mean compounds which can have a toxic effect, and they will have a negative impact on the plant, on the health of plant and or animal. And we need to know the quantity of these toxic compounds also in seaweed if we wanted to use them for any kind of application. And one of the specific compounds in seaweed, and brown seaweed and sargassum in particular, is arsenic. Arsenic is quite a well-known toxic compound, and unfortunately, brown seaweed and sargassum in particular have the ability to store the arsenic that is very diluted in the oceans, but these algae have the capacity to be able to concentrate the arsenic into the algae. And what we have also been interested in doing in SARTRAC is, okay, we know the composition of the seaweed, but how the composition is going to change when you are processing the seaweed or processing the biomass. And in particular, when you want to do something with the biomass, you need to store it. You need to do some kind of extraction. And for instance, when you are storing it, you want to dry it. And in particular, in the context of SARTRAC, because we are working with countries in the Caribbean and Western Africa, what they want to do, they want to keep the algae by drying it. At what is the influence of sun drying on the composition of the seaweed? And this is what we have been also looking at during the SARTRAC project. And so I was talking about the analysis that we are doing in the lab to look at the composition of sargassum, but the analysis that we are doing are very small scale, I will say. It's about, we are using 1 gram or a few milligrams of dry biomass. But when you wanted to obtain products from seaweed and sargassum in particular, you need to process tons and hundreds of tons of biomass. So the scale of what we are doing in the lab and what is done in industries is very different. And in industries, they are going to use different techniques to be able to extract different type of compounds. And these techniques, they will be, for instance, using liquids or using what we call solvents to try to dissolve and extract compounds from seaweed biomass. And these solvents, they can be water, but they can be other type of liquid. The biomass also can be heated at a different temperature. The heating can happen with or without pressure, or can happen with and without oxygen, for example. So there are different processes that have been developed to deal with this biomass. And what you can do also is to, after you have to, based on these techniques, you are going to produce different kind of fractions. So for instance, you are going to produce gas, you are going to produce liquid, and you will have like the solid residue. And you need to be able to separate that efficiently, to try to use the different products contained in this different fraction to be able to valorize the seaweed biomass. So this is what I wanted to say on the seaweed and the potential valorization of seaweed. On that, I will thank you for your attention. Thank you.